Back to our webinar series. Our webinar today will discuss genomics and its implications for brain tumors. My name is Jill Ann Demas. I'm Senior Program Manager here at the American Brain Tumor Association, and I'm delighted to introduce our speaker today, Dr. Will Parsons. Dr. Parsons is a pediatric neuro-oncologist and the co-director of the Neuro-Oncology program at the Cancer Genetics and Genomics program at Texas Children's Cancer Center, Baylor College of Medicine. His research focuses on the clinical application of genomic technologies in pediatric cancer care. He was fortunate to receive an ABTA Basic Research Fellowship in 2007, which supported his early research on the genomics of glioblastoma and medulloblastoma. Thank you so much for joining us, Dr. Parsons. You may now begin your presentation.
Thank you for the very nice introduction. I'm excited to be here. So I'm going to give my talk today specifically on the topic of genomics and brain tumors. And before we get into it, I thought it'd be good to just kind of give a little bit of an overview of some of the topics that we're going to be looking at and some of the objectives we have for the talk. The overall goal, as I said, is to learn about how to use genomic testing, and specifically we're talking about gene sequencing and those sorts of new technologies, which I'll elaborate on a little bit, for patients with brain tumors, both children and adults. And there's a number of specific questions we're hoping to cover. First of all, how can we use these types of genomic tests to study the biology of brain tumors in the research arena and learn more about how certain tumors are different or the same as each other? But then secondly, how can we actually start to use these tests in the clinic? And we talk a lot about personalized medicine approaches or precision medicine approaches. But the question is, how can we actually go about starting to do that? So it's an entirely new area for everybody. Third would be, what types of findings are potentially clinically actionable for patients? Because that's obviously what we're most interested in, is trying to find things with these tests that then we can help to provide information for patients or their oncologists, or help to decide, give information about what treatments might make sense. And then finally, what are some of the practical and ethical considerations of using these sorts of tests? By nature, they're very broad tests that can provide a whole bunch of different types of information and potentially clinically relevant information, including information that's not related to cancer. And so the question is, how do we actually go about doing that for our patients? And so those are the things we're going to be talking about here today. So as everyone probably on this call knows, we still have a number of, although there's been great progress in the field of neuro-oncology, we still have a number of huge challenges. And I can summarize those in terms of examples of my pediatric brain tumor patients in 3 different ways. The first is, there's still a number of these specific types of cancers that are very hard to cure. And there's some that we do a better job of and some worse, but there's some that are really quite difficult. The second is, even those treatments that we give that can be effective at treating the cancers can obviously be very challenging for our patients, both in terms of the toxicities or side effects that they have at the time of the treatments, but then also later on down the road. So this is an example of an ependymoma here. The white, we call it the enhancing portion of this tumor. You can see that is the tumor itself. And so that's a tumor that's, in theory, it's curable with surgical resection and potentially addition of radiation therapy. But you can imagine how obviously it would be very challenging and difficult to take this sort of tumor out. It can be done, but it comes at a decent cost for our patients. The third is, we would really like to know about these tumors earlier, so before they grow and cause a lot of symptoms, so that we can kind of work on more preventative and treatment approaches. So in summary, we still have a long way to go, and we're hoping to use genetics and what we learn about how tumors form and their biology as one of the approaches to improving this. So this is a summary slide basically showing schematically an example of how we think of cancers forming. And when we say cancer is a genetic disease, I don't mean that it's necessarily in all cases, or even in most cases, inherited directly through the genes that one inherits from their parents. What we do mean is that there's a sequence of events or mutations or changes that take place in a normal cell of the body that eventually lead to it having the capacity to become a tumor and metastasize and grow and cause all sorts of problems. So this diagram shows essentially on the left, and this is for the best example that we have of this is not a brain tumor example. It's been really clearly delineated in colorectal cancer, colon cancers in this case. A case how you start with what a normal epithelial cell or a normal cell of the body, and then each of these items on the top, this APC and beta-catenin or KRAS or 18q, these each represent different genetic events or different genetic changes or mutations that happen in that particular tumor cell that allows it to go from a cell with normal biology on through different stages of becoming a cancer until it's metastatic. And so when I say cancer is a genetic disease, I mean more cancer is a disease of the genes. And so just to talk about two terms, genetics and genomics are two different terms that are used in this context often almost interchangeably. And they're really just referred to the scale of things. So genetics is the study of individual genes or single genes and what they mean for biology. Genomics would be the study of many of them or all of them. And the reason that this has become relevant in recent years is that there's a number of technologies that allow us to test huge numbers of genes in tumors or in patients' bloods, not just one or not just several, but hundreds and thousands. And so that's what we've been working towards, and that's the type of information that we're trying to use. So there's a couple other points that I'd make about cancers. One is that it's a disease of mutations in genes, and this is quite complicated. So it's not the case that a tumor of a different specific type has the same mutation or the same genes or the same biology that's important, but different tumors and different patients' tumors are different in some ways. And what we try to do is learn about how to make inferences or understand what that means for individual patients. So this slide just shows an example of a representation of all the mutations in different genes that are found in a particular type of cancer. And each one of these dots represents a different gene. And the height of the peak on the mountain on one of those genes is representative of how common that particular gene is and how relevant it is for a particular tumor type. And then you line up all the chromosomes for this sort of picture, starting from chromosome 1 and going through all through a person's DNA to the end. And so what you can see here basically from this picture is there's several of these genes, these mountains so to speak, that we know are really important in cancers, and they occur in a large proportion of cancers, and those are obviously very important areas of research. But what we see is actually that although there's some of those, there are many more of these smaller cases where an individual gene might be only mutated in a small percentage of patients with brain tumors, for example. So in 1% or 3% or only a couple cases. And what we really need to learn about is how to use not only the common things, so that occur in these mountains, so to speak, but also the less common, and that's where we can hopefully define individual patients' tumors. The way we try and make sense of this in terms of getting treatments is that we try to simplify it. So instead of a particular mutation, we're often talking about a particular type of biology that we think is important in the cancer that then we can design a drug to. So this is an example of what we call over here a diagram showing a molecular pathway or a kind of signaling pathway. So this is a pathway that the cell uses to tell it when to grow and what to do. So this line represents the cell membrane. So on the bottom is the inside of the cell, and the top is the outside. So some signal comes from the outside through these genes here, and then goes through each one of these different genes, and they activate each other, and each downstream causes the signal to keep on going, to then cause the cell to have some action such as to divide or to grow or to do something. What we found is that we don't necessarily need to be thinking about mutations or any individual mutation in one of these genes, but really, for example, that in this case, mutations in any of a number of different genes all in this one pathway could have a similar biologic effect of turning on the cell in some way. And so we don't have to design necessarily a drug to each one of these individual targets, so to speak, but if we design something that it shut down this circuit or this pathway in general, that might be effective in patients. And so we're trying to take what's quite complex and for practical reasons and the drugs and treatments that are available, make it into something simpler. And that's one of our big challenges. The other thing that I would say is that although in most cases cancers are not inherited, meaning there wasn't something inherited from one's parents that directly caused that cancer to occur, it does happen in some cases. And obviously we know that happening in adults, some types of cancers, some of the breast and ovarian cancer genes, for example. But it can also happen in children. And so this is an example of a case where a single patient had a pediatric cancer, And then each of these circles or squares represent other family members, and anywhere there's color basically means that there's a cancer there. And so what I'm trying to make the point that we're trying to learn about the changes that are present in the tumors and also that inherited changes that might have produced those cancers. So how do we look at these types of changes in genes or mutations and figure out what they mean? So there's been another number of methods in recent years identified for gene sequencing or DNA sequencing or RNA sequencing. There's a million different names: whole exome sequencing, whole genome sequencing, etc. But really what they're all referring to is different ways to figure out what's the sequence of the DNA or the RNA or the genetic material in a particular patient or tumor. And 25 years ago, or even 10 years ago, this would be a bit of a fishing expedition. So there's roughly 20,000 genes, and if we were interested in any one, you could spend a career studying that one gene, looking for mutations, trying to figure out what it means in terms of the biology. And the biology part is still takes a lot of time. And so you'd have to have some candidate. You'd say, all right, well, I'm interested in this one, and I'm going to study it. But now that there are sequencing technologies that allow tens or thousands or the entire genome, meaning all of a person's DNA, to be looked at, we can do this in a much more high throughput way. So we don't have to be looking for one fish, using one line at a time, so to speak, but we can actually be doing it in a much larger way. So there's a number of different methods and tests, and this is just an example of some of them. The details are not specifically that important. So there's some like single gene sequencing, which what we would have done in the past, to targeted panel sequencing, and these are tests, for example, that might be studying 50 genes or 100 genes. These are types of tests that are used clinically sometimes in cases where there's a number of genes that are known to be relevant for a particular disease. And then you can go up to whole exome sequencing, which basically is all the 20,000 genes encoding proteins in the body. And that's the technology that most of what I'm going to talk about today is relevant to. You can also have whole genome sequencing, where you're sequencing all the DNA, not just the parts that are specifically coding for proteins. You can do RNA sequencing, and the list goes on and on and on. But so the basic principles are, we're trying to learn about how to use all these types of methods to take care of patients and learn about their tumors. So this is not drawn to scale, because if it was, the red line would go way, way off the right side of the slide. But this just kind of shows the progress that's changed over the last 10, 15 years. So before 2003 and before some of these new methods, as I said, looking at one gene or analyzing one gene in a patient would be quite a challenge. Then, in the earlier 2000s, there were methods such that you could look at families of genes or sets of genes, and then once you got up to 2006, this is where exome sequencing or whole exome sequencing and some of those methods came into play. And then more recently, whole genome sequencing, the scale of which, as I said, is off the screen. So the general point is that we now have technologies to identify far more information than we ever have before. The challenge now is figuring out what to do with that information. So these sort of methods have been used to analyze all sorts of tumors and identify new genes that are occurring in particular tumor types, which then can lead to biologic studies and clinical studies of how those genes might be used. And there are a million cases of that. I show this one for the type of tumor that I've studied in the past, medulloblastoma, that I did with this large group of people. It takes a real team approach to do this sort of work. And this is a good example because this is actually one of the projects that was partially funded by the ABTA that I'm very grateful for. And this has led to the sort of clinical application that we're now studying today. So this is a picture of a patient, an MRI from a patient with a medulloblastoma. This is the tumor back here, this white thing. This is what it looks like under the microscope. And then this just shows lists, and each one of these is a different gene that's found to be recurrently mutated in patients, more than just once. And some of them are ones we knew, but some of them, like for example these two in red, these MLL2 and 3 genes, which have a particular function of how they structure DNA and how they allow gene regulation to occur, weren't known to have anything to do with cancer, and specifically nothing to do with brain tumors. But now it pointed out an area that now we've gone on to study and trying to figure out how to utilize this information, not just for diagnosis, but for designing better treatments. So on the research side, these sorts of methods have been used for a long time. What we're more interested in doing now is trying to figure out how to actually take these same sorts of tests and use them in the clinic. And there are a number of ways we think this might be useful. First is, beyond just the histology or what the tumor looks like under the microscope, it can actually help to define what the diagnosis is. So in terms of individual tumor types, if there's confusion between different cancers based on what it looks like under the microscope. In addition, we now know that there are many subtypes of each of these tumors, so there's not just one biology of medulloblastoma, for example, but different types of medulloblastoma that have different circuits activated and in theory might be treated in different ways. This allows us to define that for patients. We can then use that hopefully to provide better information about prognosis and the expected outcomes and responses to therapy for our patients and their families, that we can decide what the best treatments might be or whether more or less treatment or a particular type of medicine might work well for patients. And then also so that we can use this information to figure out in terms of clinical trials and prioritizing the many interesting biologic findings for further study and study in our patients and using them for treatments, how we go about doing that and how we go about picking those. So there are many types of clinical applications which can be used. Going back to the example of medulloblastoma, for example, this is one example here just schematically. I'm just making the point of how this information can be useful. So this is describing studies that were published a handful of years ago by Michael Taylor, Toronto Sick Kids and his group. And what this shows essentially is at the top here is an analysis, a molecular analysis. This is actually expression profiling, so it's a certain type of genomic study for a number of patients that could be used to categorize them in different groups. So these are all patients with medulloblastoma. Each of the columns here is a different patient, and the red and the blue shows kind of what genes are turned on and turned off. And the point is that by doing this sort of analysis, instead of just having one disease, medulloblastoma, which has, certain characteristics, there's specific biologic subgroups. So this group that's marked in blue or purple here, called the Wnt. The Wnt wingless signaling pathway is a particular biologic pathway that's important, or the hedgehog pathway, or these others that are not as creatively named because we're still figuring out what they do. But the point is, we can learn how these tumors have different biology, so all medulloblastomas are not the same. This can also have direct implications, not just for designing treatments for each of these groups, but also in terms of the information and provided strategies we take. Does this, for example, this curve here, the y-axis or the up and down is the probability of survival for a patient, and the other axis moving from left to right is how long since diagnosis. So if we start here, it's patients initially diagnosed, 100% of them survive, and then over time, obviously some of our patients have problems related to their tumor. But what you can see is that by the different colors of these different groups, there are very different outcomes. So for example, this blue line, which is the Wnt pathway patients, the prognosis is really very good. I mean, given the circumstances and given the treatments that we have to give in chemotherapy and radiation therapy. But so those patients, virtually all of them, do very well in terms of being cured of their tumor. Whereas some of the other groups, it's very, very different. And so obviously these are the patients where we need to think about different strategies or more aggressive treatments or different clinical trials. And so this type of information can really help us provide information for our families and our fellow oncologists about how to take care of the patients. And there's also, obviously, the dream is that we can then design specific drugs to use for these specific subgroups of patients. And so this is examples, again, published about 5 years ago for the disease medulloblastoma. It was actually an adult with medulloblastoma. And it was a medulloblastoma that had spread outside of the brain and central nervous system throughout the body. And it was treated with a specific pathway inhibitor targeting this hedgehog pathway, which was one of the biologic aspects which we just mentioned. So this is the before, and each of the black bits on this are places where tumor has spread. So it's really been all through the skeleton, and this is the spine, and then the shoulders, and the pelvis, and the hip bones. This panel shows what happened after treatment with this single inhibitor. So not other chemotherapy or not other radiation therapy. Basically, all of these dots and all of these black bits are gone. And this is the heart, and that's the bladder, and that's normal to have that light up. But basically, the tumor went entirely away temporarily. But then the challenge is, even if we're finding medicines that work, we have to figure out how to use them in combination with other treatments, just like we do for other cancers. Because this treatment alone, the tumor started to come back. And you can see how there's the black dots here again. And so that's what we're trying to do. Find drugs that will improve the outcomes for patients and then figure out how to use them with other therapies. So that's the overall goal of using genomics for personalized cancer care, to try and take these methods that I've been talking about, whether they be exome sequencing or genome sequencing, and use them in a way such that we can actually take care of patients and help take care of our patients and their families. And so instead of a patient having surgery and then being diagnosed with a particular tumor type like medulloblastoma and then all getting the same treatment, that hopefully by our knowledge of the genes and the biology, we can decide which patients need what treatment and what treatments they might best get. So that's the dream and the promise of personalized medicine. The rest of the talk, I'm going to be talking about ways in which we're trying to study this process and overcome these challenges and what kind of information we can get from this sort of approach. So this is a list of some of the challenges to doing that. So the challenge is to be able to take genetic information and then use it to take care of patients. And it's a long list. First of all, we don't understand what many of the mutations and much of the biology that we find in these tumors. So it's a very great area, an active area of clinical research that we can, or basic research, that we can find some gene that's been mutated in a type of brain tumor and then study what that means in various models and cell cultures and all that sort of thing. But in terms of direct applicability to patients, there's obviously some lag time in there while we learn what it means. The second is there's a relatively limited number of preclinical models for brain tumors, so cell lines and various other types of models that we can then test therapies or test the biology of these things. The third is all this information about the specific genes and targets in cancers is relatively recent, much of it. And so drug companies are rapidly developing agents and drugs to use for these purposes, and there's a ever-increasing number of these. But the fact of the matter is it's still an area of growth. So we're still acquiring a number of drugs. So even if we find a particular mutation in a gene, oftentimes there's not a drug available to target that yet. And so that's what we're working to improve. Fourth is we're just not used to using these sort of tests in the clinic. So these broad genomic tests that can give you all sorts of information about tumors and why tumors and other diseases might have developed. And so much of what I'm going to talk about in the rest of the talk is our experience here at Texas Children's Hospital and Baylor College of Medicine of doing a study of this type in this type of setting for our patients and what we're learning from it. The last is there's a lot of challenges of clinical trial design. So it's great to say that medulloblastoma, for example, is not one disease, but it's a number of different diseases and we're going to treat them all in different ways. But what that means then is that we have to do clinical trials where we separate them out in all those different subtypes of medulloblastoma and then try and prove using our drugs and clinical trials that we can improve care for those patients. So then we get into even smaller numbers, so if you're talking about very specific biologic or molecular subtypes of tumors. The other part I would say is oftentimes for patients whose brain tumors come back, there are not biopsies done. And so getting material to use to dictate what treatments a patient might get can be a challenge. So we could come up with a longer list, but that's the one that we talked about right here, kind of a summary. And so the rest of the study, I'm going to be talking about a particular clinical study that we're doing here. And it's an example of the ways in which researchers and cancer doctors are trying to learn more about how to use this process. It's a study that we call Basic 3. And this is my co-PI of the study here, Sharon Plon. She's a cancer geneticist here at Texas Children's and does cancer genetics and genomics research, similar to what I've been talking about. So the goal of this study is to take information from clinical, so CLIA certified just means in a clinical laboratory, so not in a research laboratory, germline, meaning the inherited, and tumor exome sequencing into the care of our patients. So to try and set up the process to do this clinically, so to get the testing done, to explain it to the families of our patients, to interpret the results, to figure out how to use it, that's the goal of the study. And then we're in parallel evaluating what the impact of this information is for our families and our patients and their physicians. So this isn't a study where therapies are being dictated by the results of genomics, but it's rather one where we're doing genomic studies in the clinical arena and then studying how often we think we can find useful information. So I'll just walk you quickly through kind of the format of the study. So over a couple years, we're enrolling about 280 children with either brain tumors or other non-CNS solid tumors, but a number of brain tumor patients. Blood and tumor samples are acquired from those patients. They go to the clinical lab to have this genome sequencing done. So it has, it's done just like other clinical tests, like when the blood count is done in the hospital. I mean, there's certain, quality criteria and standards for signing out the results and interpreting them. It's all done in that same way as any other clinical test. 2 reports get generated. One is the somatic, which is what we mean as the tumor-specific mutations. So this is the list of mutations that were found in that patient's cancer. The other is a germline list, which has information about why the patient might have, it's from the analysis of their blood, and why they might have developed the cancer or have risk for other diseases. And the question is, how much information of these 2 types can we find? Those results go back into the medical record. That's what we call the EMR. And then along with the patient's primary oncologist and a genetic counselor, which is GC, those results are actually given back to the family and discussed with the family, and then any appropriate follow-up in terms of interventions, whether it be referral to another clinic or whether it be a particular treatment, can be taken. So the idea of this study is, and then we follow the patients for several years after that to try to see what decisions get made based upon this information and what the outcomes of the patients are. And so this is a study of kind of the practicalities of doing genomics in the clinic and learning about the process and learning about how we can best try and do this for our patients. So the rest of what I'm going to be talking about are some of our preliminary results and the examples of what we can learn from this type of study, which I hope will be interesting. So we're more than halfway through the study so far. There's 150 patients enrolled. We also enroll the parents of our patients, not because they have all the genome testing done, but because they are interviewed and surveyed and actually also provide blood to interpret their child's testing, if they're so willing. All of our pediatric oncologists, and this would be the similar sort of study, it could be done on adults or children. In this case, we're doing it in children. Here at Texas Children's are enrolled. And this pie chart just shows we're not picking specific diagnoses. We're talking about all different types of tumors. So medulloblastomas in blue, low-grade gliomas in red, ependymoma in green, etc. And in both adult and pediatric cancers, obviously, there's a pretty great diversity of these sorts of things. So we are trying to study not just one type, but all of them. So the first observation I'd make is that our families and our patients are very excited about this type of approach, which is both a blessing or an opportunity and a challenge. So more than 80% of families agree to participate in this study, this clinical study. Of the ones who choose not to participate, the majority are just because they are not interested in participating in a research study at all, that they're so overwhelmed by the new diagnosis. These are all newly diagnosed cancer patients, or they have just so busy, stressed, etc., so entirely reasonably aren't able to participate. The number who have specific objections to the study, so for example about the privacy and having genetic testing done or the blood draws, is much lower. So most of our patients and their families agree to participate. But that's the challenge then we have, is trying to make it clear to them that this is still an area of investigation and that we can't promise that there's going to be some benefit to this sort of genomic study, but we're really trying to learn whether that's true. And so that's one of the challenges, but there's pretty great enthusiasm. So this slide just shows an example of some of the practicalities, with using this type of approach. So everything has to be figured out, from how to talk to the families and patients and obtain consent, obviously, for this type of study, to, for example, how do the samples get handled. So this is an example, our newly diagnosed patients. They have surgery and study enrollment is offered. The surgery, the tumor sample goes to the clinical lab, so for the diagnosis of just as always been done. And if there's any left, there's some fresh frozen tumor, and if the patient enrolls on study, then that gets sent to the clinical lab. And if not, obviously the sample gets handled appropriately. And this is Angshmoy Roy and Dolores Lopez-Torrada, who are our molecular pathologists here who coordinate this. But just the simple fact of getting the tumors where they need to go and getting high quality samples is critical. So there's a number of research questions we try and answer in this sort of study. The first is, number one, just kind of some of the clinical questions associated with this. So for example, the goals of providing both the tumor results, so what can we learn about that cancer and what its response might be to different treatments, and then also about the blood and results, or the germline results, which I'll also talk about just a little bit. So most of what we're going to be talking about here is the tumor genomic results and how those might be applicable to patients, in particular those patients whose tumors come back and need additional treatments. So we try and look at how important the mutations we find are in a number of ways. First of all, we do actually observe what the oncologists do for their patients. So for example, if we find a mutation based on this type of testing, do they actually use it to make any decisions about what drugs to give their patients? We also survey and interview the oncologists and the families at various time points. But the other thing we do is the list of mutations that get identified in a patient, and they've ranged from none in their tumor to up to around 80 in the exome test. They get categorized based upon how clinically applicable they think they're going to be. So ranging from category 1 mutations are ones that are of some established clinical utility. So there's something that should be done with that test result, whether it means there's information about prognosis or a specific therapy. But these are the things that get tested for anyway, basically. The category 2 is the much more interesting category of potentially clinically relevant mutations. And those are cases where there might be experience with targeting a particular gene. So for example, this gene MET is a commonly mutated gene in many tumor types, but it's not commonly known to be in this particular tumor, neuroblastoma. And there might be something that the oncologist might say, wow, I think this drug potentially could be of use for this patient, but there's not a lot of data. And so that's where we're trying to learn about how to use this information. Third, there can be mutations in other genes that have been associated with cancer, but that we don't have, clinical applicability of. And finally, in gray here are kind of the all other mutations. So ones where we don't, at the current time, know almost anything about what they mean for that patient's cancer. So there's all different types of mutations. And the one thing I'd show is that if you can see, the majority of this slide is gray. Most of the mutations that get found in patients' tumors, we don't know what their relation is to cancer. So this shows each row here is a different patient, and this is different tumor types, but they're all just lined up from most mutations we found to least mutations. And then the height of the bar is how many mutations were found in that patient's tumor. You see it ranges from about 80 all the way down to 0. And the vast majority of these, as I said, are gray, meaning additional research is going to be necessary to figure out what they mean for patients. But you can see there's a little bit of yellow and red and orange on these different patients. And those are cases where there is something that's potentially clinically relevant. And that's what we're focusing on right now in terms of direct applicability. This is another way of looking at that. So from an individual patient level, how often do we find mutations that have different levels of potential clinical relevance? And the color coding is the same as on the previous slide. So for example, about half of patients here in gray don't have mutations in any genes that we currently understand to be related to cancer. And so that's obviously a key area for basic and future research. But then about half of them do have mutations that are either of direct clinical utility, potential clinical utility, or in other cancer genes that we're still trying to learn how to use it. So, and this is from one test. The question is going to be for different types of tests, and combining different types of tests, can we make this red, orange, and yellow bars increase and increase and increase to where we're providing better information for most patients. This is just a list of some of the genes, and it's not specifically important, but the point is that there are some genes that we see that are frequently mutated. This is a gene called beta-catenin that's important in a variety of brain tumors, p53, etc. And so those are obviously clinically interesting. But again, there's many important genes that we only find mutated in, for example, 1 out of 80. This is the data for the first 80 cases. 1 out of 80 cases. So for example, there are inhibitors of ALK. There are inhibitors of the RAS-RAF pathway, so this mutation called NRAS, etc. And so this is something that there are interesting findings here. And this is just one specific example of one of those cases. So it's a 14-year-old boy. He had a tumor in his cerebellum, this here in the white here. Under the microscope, it looked like medulloblastoma, and that was the diagnosis. The pathologist, and this is from Angsham Roy, one of our molecular pathologists I showed previously, the pathologists then do other stains in addition to this basic analysis of the tumor under the microscope to try and look and see what biologic subtype they think it is and that sort of thing. And those results in this patient were just fairly a little bit confusing. So then we did the tumor test, and there's a number of different results that are potentially interesting in this patient. Each one of these blocks is an analysis of a different gene. So there's 1, 2, 3, 4, 5 of them that I'm showing in this slide. In each case, the normal sequence, so what's present in the patient's blood, is on the top. And this is just a read of the DNA, so to speak. And here you don't see any changes, you don't see any letters, everything's normal. Whereas in the tumor, you can see some cases where there's a different letter here, and those are cases where there's a mutation or something changed in that tumor. So in this case, there was one mutation in a gene called CTNNB1, which is beta-catenin, and also another in a gene called DDX3X. And so these define this patient's tumor as being in a specific molecular subtype of medulloblastoma, the Wnt pathway that we talked about. And those patients have an improved prognosis, and this is actually important information to know potentially for how to talk with your families and patients. In addition, then, there are mutations in a gene called P53, which is probably the most famous cancer gene of all, which there are also is some information for some patients about prognosis, and there's also attempts in various clinical trials to try and target pathways related to P53. And there are also mutations in genes called ARID1B and FOXO3, and these are each cases where the biologic pathway or function that these genes are in are ones that are kind of the targets of investigational agents. So by doing this sort of testing, we can see not just confirming some more about the molecular diagnosis and what subtype of a tumor, but also things that are potentially relevant to future therapies. And so that's the sort of thing we're trying to do. So we have a diagnosis here now of a Wnt subgroup medulloblastoma. The prognosis is for medulloblastoma standards quite good, and there's some clues about specific genes that might be relevant in that patient's tumor. The other thing that we try and do here, as I emphasized before, the exome sequencing is the one genomic test that's being done, but we're also trying to do all sorts of other tests to learn what is the exact, in the end, what are the exact methods that we can use to find the most useful information. So looking at the RNA, looking at not just the exome, which is a part of everyone's DNA, but the whole thing, the whole genome sequencing, etc. And so our patient families all specifically give us permission to do some of these other studies, which is very nice. And we're working to learn more about the tumors. So up to this point, I've been talking entirely about the tumor and what we find in the mutations. The other thing that I would say that we're working pretty actively on is trying to figure out more about what these mutations mean. So we have a list of mutations in a patient's tumor. We might have ideas about what that means for treatment, but the question is that actually useful? And if you targeted, for example, one of those genes that's mutated in that tumor, would it actually do anything? So there's all sorts of methods we're using to develop model systems, and this is Xiaonan Li, who's a researcher at Texas Children's. He basically developed these models of brain tumors by using tumors, and this is the dark blue here is a bit of tumor that are injected into mice. So they can take the human tumor, which has all the biology of the human tumor, put it in the mouse, and then in cell lines and in these sort of models, learn more about the biology, and then also test ways to test particular treatment approaches or drugs or immune therapies that might best do this, so before it gets to the patient. So even though we have lists of mutations, we really then need to learn about what we're doing with preclinical models. So far I've been talking entirely about the tumor. In the last couple slides, we'll point out that there are other considerations here which actually get a lot of attention and a lot of questions that people have. And this is, we're also, by sequencing the blood of the patient, we're trying to provide germline or inherited test results for the families. So we can see, for example, if there's risk factors there that explains or helps to explain why the patient developed that cancer, whether they've inherited, for example, a mutation in a gene that predisposes patients to brain tumors and other cancers. And that obviously can have potential impact for genetic testing with other family members, for surveillance strategies, for example, doing MRI scans or some sort of screening scans in patients who we know are at risk of developing those tumors but haven't yet developed them, so we can hopefully diagnose early. In addition, we're interested in learning how often do we find results that are clinically important for patients but not related to cancer, so to other diseases that are not cancer. So this is an example of one of those cases of inherited cancer mutations. So we had a patient, a 14-year-old, who had a brain tumor, and in our medical record there was no evidence in the notes of any family history of cancer. So the blue marks someone with the cancer, and the father and the mother and the other siblings. However, when we talk more to the family about this, there is actually cancer, it's just not directly in the mother, father, or siblings. And so it was suspicious for concern about some familial risk family for developing cancer. And so when we sequenced the patient's blood, we were able to find a mutation in a particular gene called MSH2, which is one that's known to be a risk factor for increasing the risk of multiple types of cancers, including brain tumors such as gliomas, which this patient had. So this is a case where by identifying these sorts of results, it actually has implications both potentially in some cases for the treatment of the patient's tumor, but also for other family members who might or might not have these sorts of results. Because as I said, one of the things we're trying to do is not just treat brain tumors, but prevent them or diagnose them early when we can. So the results can also be related to inherited risk of cancer. And this is a slide that shows just schematically all the other types of results you can get from a test like exome sequencing. So for example, this is on the left the tumor report. Basically all the mutations that are present in that patient's tumor get reported by the lab. This is the blood or germline report, where you can get pathogenic, that means disease-causing mutations related to cancer, or if the patient has some other disease also related to that. But you also get what we call VUS, which are variants of uncertain significance. So these are changes that are in that patient's DNA, and they're in a cancer gene, for example, but we don't know how to interpret them. So they're the equivalent to some of the tumor mutations that we're still learning how to figure out. And so those are there. We can also find mutations that are medically actionable in other ways, so not having to do with cancer, but for example, for saying the patient's at risk for having heart rhythm problems or having heart attacks or having seizures or something along those lines. So an example of that is this SCN5A is a sodium channel mutation that is associated with a risk of heart rhythm problems and potentially serious rhythm problems. In addition, we can find mutations in genes. PCG stands for pharmacogenomic, pharmacogenetic, mutations that tell us how the patient might metabolize drugs and whether they need higher or lower doses of certain drugs, which can also be helpful. We can also find information about that talks about the risk of being a carrier for other genetic diseases and all sorts of other things. But this is just an example showing when we do this sort of testing, we need to consider not just the tumor and not what just tumor results we might find, but what we can see in terms of other diseases and other uncertain findings as well. And so having a plan from the lab and the clinician and genetic counselors available for this type of testing can be very important. So this just shows the kind of diversity of results you can find from these sorts of things. So this shows, 1 through 34 are just a set of 34 patients of the first, say, 80 or 90 that we tested. The germline are the blood results, the tumor are the tumor results, and there's the different categories of what types of potentially clinically relevant information. And you can see there's all different dots in all these different categories, but there's a number of patients who have findings either in the tumor or in the blood that have some potential clinical relevance. And so refining this further and figuring out how to use better tests to improve the yield of these sorts of things for patients is critical. The other thing I'd say, which I haven't talked about at all, is that in addition to doing this sort of testing, we have a whole team of ethicists and social scientists who specialize in doctor-patient communication, who actually audio record some of these visits between the oncologists and the families where they explain the results, and they can try and learn what we're not teaching well, what we're not explaining well, and what types of questions families ask and how we might better kind of serve them with this testing. But then also about the preferences and attitudes of both the oncologists and the family members regarding these sort of tests and how they think they should be used and what information they're interested in using. The idea is to integrate these all kind of seamlessly into the standard of clinical care. So the other thing we're trying to do is use these things in prospective clinical trials. So the study I've been talking about here is one where it's all observational, meaning we're doing this testing, we provide the information to the oncologists, but we don't actually dictate what treatments the patients are going to get. So moving forward, the goal is to do some things that are more interventional, where we do this sort of testing, and then depending on what the test shows, patients get different therapies. But more importantly, we need to study that in clinical trials and figure out if that's an approach worth taking. And so that's what we're trying to do. The challenge becomes, as we mentioned, there are numerous practicalities. And so this is a slide when we surveyed some of our oncologists here at Texas Children's talking about, well, what different genetic pathways or what different biologic pathways do you think would be important to try and target for clinical trials for patients? And the size of each of these words represents how frequently they came up with that. So RAS, MAPK is a certain biological pathway that's drugged. There's the PI3K pathway, immunotherapies, a number of different things. But the fact of the matter is, although we have great ideas about many of these things, and some of them are in current clinical practice, many more are in early stages of investigation, meaning new drugs for each of these. We're still learning what their doses and toxicities are, and we're learning how to use them and combine them. So it's a complex process, but one that's very exciting. So in conclusion, I would just say that hopefully this was an example of some ways in which genetic and genomic approaches are helping us to learn about brain tumors and other central nervous system cancers. The second is that the results of these types of studies have real clinical applications and have the potential to lead to improvements in patient care. But we're still learning about the process, and so there's some testing that can be done, but it's nothing like what it'll be in 5 to 10 years, where we can get in addition to getting a great deal of information about why tumors have developed and what their sensitivities might be, but we can actually figure out better ways to implement that and use it for our patients. So this is a slide just showing a small representation of the many people who are involved in that basic 3 study that I was talking about, starting with Sharon Plon, who's my co-PI or co-leading the study with me, and a million other people, from pathologists to surgeons and neurosurgeons to basic scientists to genome sequencers, et cetera, in a number of different places. So it's doing this sort of things in the clinic requires a real team approach. The last thing I would say is that a big thank you to the ABTA. So this is an example of further follow-up research in ways that research was funded through a basic research fellowship when I was first in the laboratory studying in the lab several types of brain tumors, has now evolved into not just further studies of those brain tumors, but more clinical studies and trials and analyses that hopefully will help patients. So I'm very grateful to the ABTA for that. And then last but not least, I should mention thank you to our patients who participate in this with us and who let us take care of them. And I will stop there and we can take questions.
Thank you very much. That was very educational, Dr. Parsons. So Dr. Parsons will now take questions. If you have a question you'd like to ask, please type and submit it using the question box in the webinar control panel on the right-hand side of the screen. And we have a few that were already sent ahead of time, so I'll start with some of those. Someone is asking about that they have a family history, melanoma and prostate, the strong suspicion that the genes predispose them to tumors. So what does someone do when they think that there is this running in their family? How would they even start the process to know if they're predisposed? Where would they reach out to?
Yeah, so that's where they need to take advantage of their oncologist. So although the oncologists, and I'm one of the oncologists, are not specifically trained necessarily in genetics and inheritance of genetic diseases and these sorts of things, what they can do is help take an initial kind of very careful family history. Often we're hurrying, we're trying to figure out what treatments we're going to give, when the patient's going to go to the operating room, asking some really important questions from the family about other family members of which types of cancers, how old they were when they had them, and getting that information to the oncologist so that then with the patient they can decide whether they need to refer to a cancer geneticist or get some additional thinking on that. But so providing that information and having some good information on various family members, and not just, as most of us are, kind of a vague sense that, oh, there was some cancer there back in my grandma's generation, but actual having data that you can provide so that they can assess the risk of cancer is very, very helpful.
Great. And you mentioned 2 different parts, that genetic counselors were an important part, and then at the very end you talked about the ethical considerations. Can you talk about how those work together? Because people are interested in the fallout that happens when you do find out these results, that there may be these things in their genes. How do the counselors work together with the families to kind of minimize the worry that's going to happen when they find out that there's all these possibilities that could happen to them?
Right. So I guess a couple points I make. Number one is that we have a pretty high bar for considering things medically actionable, meaning we're not reporting from these sort of studies that someone, might have an increased risk of diabetes when they're an older adult or, those sort of things. It's more cases where if I was the doctor for this patient and I knew this particular result from the testing, there would be something I'd be supposed to do for my patient, whether it be referring them to another specialist or doing some sort of testing or giving them a treatment. So in this case, we're lucky because we're studying kind of the best case scenario here, which is a case where it's kind of one academic group. We have two fantastic genetic counselors who work with us. The primary oncologists are also involved in the study, and so the most important part is we have this team approach. We have access to the genetic counselors, so if there are questions that the oncologist is not as comfortable asking or doesn't have answering or doesn't have time to answer, they can help. But then the most important part, I think, is we're not just in the business here of providing information, hopefully, but providing information and then helping our patients do what needs to be done next. So not just telling them you have a risk of this or that, but saying, here's the finding, here's what we think the risk is, and here's the other specialists or the other doctor or the other tests that we should do to help take care of this. So I think that is pretty effective, hopefully, at streamlining it and reducing the anxiety. And our experience has been that that's been effective.
That sounds wonderful. Someone's asking a question, if sequencing could help find a specific site where a drug could attach to the gene to shrink the tumor?
So that's a great question, and that's the sort of thing that we're trying to do. So for example, there are specific mutations in certain genes that we know what it does to that gene and the protein it codes. So it alters the structure of the protein somehow. It makes it so that it doesn't bind properly to to some other protein. So there's cases where we have a pretty good idea of what the effect of the mutation is. And so in those cases, I'm using the we very loosely, drug companies and researchers are trying to design drugs that could target that specific structure or that specific shape in exactly the way you're talking about. And so there's some cases where that's been shown to be effective. There's, for example, inhibitors of a gene called BRAF or ALK, or some cases where that's been an effective approach. So far, they're more kind of proof of principle or examples and not something that we can routinely do across the board, but that's our goal.
Nice. It's exciting to think that can happen soon. And where can patients expect to go? Is it happening, only at teaching institutions? Is this happening, just at a half a dozen places, like your institution?
Yeah, so it's something that's happening more and more, and using some of these sort of tests. So not necessarily as part of a big trial, or not necessarily using like the exome sequencing test, but using some of these tests to look at genes are being done more and more routinely in both adult and pediatric oncology everywhere. What we're trying to do, and the advantage to doing it kind of in a larger group or a place where we have kind of all the different specialists who are involved in this, is to do more to study like the process of how effective that can be and better ways to do it. But in terms of this type of testing, it's something that's becoming more and more frequent, but the challenge is that most often the results currently are very difficult to interpret. And although there might be something that's biologically interesting in the results, that most often there's not something that's directly relevant to picking medicines.
Do you then worry that if the results are difficult to interpret, that someone's not reading them that should be reading them, or is that a concern?
Yeah, it's a good question. So I do worry that the results, what we worry most about is that the results can some way be overinterpreted. Like, that's the challenge, right? That the labs are trying to report these things, they're trying to explain what they see in the tumor, but then it's the oncologist or the team taking care of that patient who then has to interpret it and decide, it's just like, seeing a result of an MRI scan or other testing, then decide what to do about it. Mm-hmm. And that's the part that's still a little bit more of a gray area, and that's the part that's both a challenge and exciting.
Absolutely. So definitely more to come and exciting for the future. So it looks like that's about all the questions and all the time we have. So I want to thank you again for the presentation as well as answering all the questions. For more information on brain tumors and other topics, we want to refer you back to our website, which is abta.org. Also encourage you, if you have any further questions that we didn't answer today, to call our 800 number, 800-886-2282. You can talk to our healthcare professionals here. So if we can pause just for a moment, and we're going to stop our recording.